better during commercialization steps. So I would put efforts in both, however, make emphasis that to do as much possible as for alternative methods.
Brilliant, thank you. Lucia, what challenges are you still facing in your work that you hope to overcome in the next year?
Yeah, I think we and also quite a lot of other companies still face the issues with reaching enough high yields. And this is also what our development work is also focused on, so to increase the yields. In addition, we face also, I would say, problems with full empty ratio. So in the next year or the next time, we're also focusing on increasing the full empty, so the portion of the full capsids in our product.
Excellent, thank you. And then Alina, I'd ask you the same question. What challenges are you facing currently?
So as a new platform, any type of new platform, you are facing something which was around and regulatory agency get used to for decades. So the biggest challenge is mentality shift, is cultural shift to put different perspective. So you need to understand that before moving forward with something, and first time you will hear no, potentially it's not no to your platform or to your method. It's not to based on this developed previously for previous platform. So you need to invest time and make your historian search, and then change a strategy to explain in highlight of historic issues in order to get yes finally for alternative method or some analytical strategy for the new platforms.
Excellent, thank you. And then Lucia, if I go back to you and ask you, what would be the key take-home points from your presentation today?
Well, that our platform at Takeda is really a good one, I would say. So what is important for gene therapy in general, and also with respect to us, that the process is really good from the beginning, so starting already with phase 1, because it is, I would say, very hard or complicated if you improve your process in later phases, and then you need maybe, or you have a problems with comparabilities, and yeah, so this would be the main.
Excellent, thank you. And then Alina, if I could ask you the same question, what would be the key take-homes from your talk?
I would say that no matter how sometimes frustrating it seems to start with new technology, and it shouldn't be discouraged, and also keep in mind that there are a lot of background work from different platforms in order to shift this mentality. So something for, I made an example for recombinant technology, which start from digital information, but it took years to challenge everything around. So platform is ready, but surrounding regulatory environment, it's not. It didn't discourage us. It was a hard work, but it's paved the way the other digital-based platforms around on the market, like DNA vaccines or mRNA, which basically also kind of new one, but surrounded by the same regulatory environment. So that's what be the take-home message: not discourage, and keep in mind that it will pave the way to multiple new development in the field.
Excellent. Lucia, if I go back to you, in the wider industry, what do you think is really exciting at the moment?
Excuse me, I didn't get your question.
In the sort of wider industry as a whole, what sort of areas do you think are really exciting right now?
Yeah, I would say maybe the possibilities to maybe switch from plasmids we are currently producing in E. coli, maybe switching to synthetic plasmid production. This would be a great
excellent. And then Alina, what's from your perspective is the most exciting parts of the industry right now?
Hello, excuse me.
From your perspective, what are the most exciting parts of the industry?
I believe I lost connection for a little, but I assume it is a similar question.
Yes.
Yes.
What are you finding most exciting?
So I would say that all industry, it's moving like with pace of digitalization, and this is something to keep in mind. And everything turned out that there are technology already there, and commercial one like recombinant, when you need to start production of something vaccine against infectious virus using digital file, so which could be sent by email, right? And so this is something like breakthrough. And while the entire industry, not only biopharmaceutical, moved over digital, so I think that overall that will be the path forward, that the possibility and opportunity to exchange information in the field with the speed of how digital platform could make it.
Excellent. And then to follow up, we just received a question through from the audience online, and that's Alina, you mentioned earlier planning of high throughput assay in the PD pipeline. What's your opinion on automated cell functional assays? Is Sanofi going to be interested to see such off-the-shelf automated cell assays customized to your current assay setup?
Well, this question will probably be better suited for our R&D side. Obviously, we are always interested in something which is make it more high throughput, more advanced, and we try to make as much customizable as possible. And our R&D research and development are really made a lot of effort from very beginning to make it to put emphasis on automation. So when something which became commercial, well, and turn into industrial affairs, then it will really make a huge impact.
Excellent. Well, thank you very much, both. That looks like all the questions we've had through right now. I'm sure if there's any further questions, though, do reach out to either of our speakers. I'm sure they'd be happy to discuss further. But thank you both very much for your presentations and your time in answering these questions. And we hope everyone continues to enjoy the conference for the rest of today.
Thank you.
Thank you very much.
Thank you.